Recording. All right. Ominous warning about recording here. So welcome everybody to the second remote Boston Protein Design and Modeling Club. I'm really excited to introduce our speaker tonight, Brahm Yachnin. Brahm did his postdoc with Sagar Kare at Rutgers, and now he's in Boston. So our Rosetta community grows. So welcome, Brahm, and the floor is yours.
Yeah, so as Chris mentioned, I did my postdoc with Sagar Kare, and I'm now a scientist at Vistera out here in Waltham. Unfortunately, I don't have anything about the stuff that I'm working on right now that's ready to share with you guys yet, though hopefully that will change sometime in the next, let's say, year or so, depending on what happens with all this pandemic stuff. But I'm going to tell you, today I'm going to tell you a couple of stories about stuff I've worked on during my postdoc. Hopefully not too many of you have seen this before, as I presented a few times, so sorry if this is stuff you've heard before. So the two stories, the first thing I'm going to tell you about is kind of a more applied story, about how we used Rosetta and some massively parallel screening methods to identify pro-enzymes for use in next generation chemotherapy. And the second thing is more of a feature that we added to Rosetta to help with the de-immunization of protein therapeutics, specifically non-antibody therapeutics. Yeah, I'll get right into it. So the therapeutic system that we were working with is directed enzyme prodrug therapy, or DEPT. And the way DEPT works, so as you guys I'm sure are aware, chemotherapy normally involves a drug that is toxic and damages not only the tumor cells, but typically all healthy dividing cells as well. And so the way DEPT works is we take an inactivating group and attach it to that drug to make it a non-toxic prodrug. And that prodrug can then be activated by an enzyme that is separate from this, from the prodrug, and it converts that non-toxic prodrug into its active drug form, triggering the toxic effects that drug possesses. The way this is useful is that we take that enzyme and we hook it up to an antibody, and that antibody is going to be directed to the tumor site. And I say antibody, but it does not necessarily have to be an antibody. There are other targeting mechanisms that have been explored. The antibody delivers the enzyme to the tumor site, and then the prodrug is administered, and that circulates through the body, arrives at the tumor site, and the enzyme then goes ahead and activates the prodrug, turning on the toxicity locally in the tumor microenvironment. So you have non-toxic prodrug going around the body and toxic drug at the tumor site. And so this should protect the patient from the toxic effects of the drug. So there's a number of reasons why this is useful. So as I just mentioned, that will reduce the side effects associated with chemotherapy. So you're going to have much fewer than those nasty side effects that people who go through chemo are familiar with. It also means that we can use higher doses of the drug or more toxic drugs than we could normally tolerate. And this is important because it means that it reduces the chances of developing drug resistance, because just like how when you're given an antibiotic, it's important to make sure you take it for the duration of how long it's prescribed, to make sure you get rid of everything and you don't allow a small remnant to remain that can develop drug resistance. The same is true with chemotherapy. If we don't give enough drug, the cells can, a small remnant can survive, develop resistance, and then the tumor comes back and doesn't get treated by the drug. So in principle, if you have higher toxic punch to the tumor, you may be able to mitigate that drug resistance. And as I alluded to before, there's a variety of ways of targeting the enzyme to the tumor site, which makes this a kind of modular approach. So you can use antibodies, as I mentioned, and that allows you to target a variety of different tumors because you can use a different antibody for every tumor type that you're interested in targeting. And you can also think about things like gene therapy, inhibitor DEPT, and substrate DEPT. So these are all things that have been explored. This is not to say that this is easy to switch your specificity from one thing to the other. It's still a lot of work, but it means that you're not starting totally from scratch and developing a new kind of therapy. So the specific system that I was working with is the carboxypeptidase G2 system, or CpG2. And so CpG2 cleaves and inactivates this drug, I should say, this drug CMDA and converts it to a nitrogen mustard drug. So that's essentially how this is going to work. This is fairly well studied. There's been a number of clinical trials from a number of years back, and this was primarily in colorectal cancer patients, and they saw successful regression of the tumor with minimal side effects. So that was pretty exciting, though in a limited clinical studies. Another nice thing about this particular system is that carboxypeptidase G2 is actually a drug right now. It's indicated for the treatment of acute methotrexate toxicity. And so this is something that you don't have to necessarily worry about all of the safety issues that might come with a totally new biologic, because we do have some track record in patients already. Okay, so I've told you why this seems like a good idea and why it has a lot of potential. I haven't told you why we haven't started using it all over the place yet. And one of the chief limitations of DEPT is the complexity of the dosing regimen. So typically with DEPT, you are dealing with two different components to your therapy. You've got the enzyme with its targeting molecule, and you've got the prodrug. And these have to be kept separate until they get to the tumor, otherwise the enzyme is going to activate the prodrug. So if you've got if you imagine your patient and you give that patient the prodrug, the enzyme is going to flow through the bloodstream and it's going to arrive at the tumor site. And if you were to give the patient the prodrug at this time, you would have systemic toxicity from the circulating enzyme and tumor toxicity from the tumor-associated enzyme. So basically, the same thing you get with standard chemotherapy. So you have to wait for the enzyme to clear out of the bloodstream so that you have no systemic toxicity, but you still have the tumor toxicity because the enzyme remains localized to the tumor. If you wait too long, on the other hand, the enzyme gets cleared off of the tumor and you get no toxicity at all. And so getting this balance right is, of course, critical, and it is also quite difficult. So the reason for that is that it varies widely from patient to patient and can be on the order of days. So the way this was resolved in the limited clinical trials that have been done is you assay the patient's blood regularly and wait until the drug level, the enzyme levels fall, and then you give the patient the drug. And while that might be workable in a limited clinical study, it's not the best way of going if you are trying to do this as like a wide-scale treatment. So that's one of the chief things that have to be overcome with this therapy. Our idea in order to deal with this is to use tumor proteases. So many tumors overexpress proteases naturally. And our idea is to use an enzyme that can be activated by those proteases in order to trigger the the activity of the enzyme and the toxic response locally at the tumor site, giving us an additional layer of regulation. So the way this would, and so we're making our enzyme into a pro-enzyme. So the way this works, we deliver, rather than the regular enzyme, we attach a prodomain to it, which inhibits the enzyme. And then when that enzyme arrives at the tumor site, you have proteases, which are overexpressed by that tumor. Those proteases go and cleave off the prodomain, reactivating the enzyme and allowing that enzyme at the tumor site to trigger the toxic effects when the prodrug comes around. So you would have inactive enzyme in circulation and active enzyme at the tumor site. And so that's what you see here. So you have no systemic toxicity in the bloodstream, and then at the tumor site, the proteases get expressed, turning on the enzyme and triggering the toxicity at the tumor site. And so in this way, you wouldn't have to worry so much about giving the prodrug too early because the activity in the bloodstream will be lower. When we first started off on this project, we looked at the structure of CpG2, and you can see that here. And actually, can you see this pointer? Is that visible? My mouse, yes?
Yeah, we can see it.
So these two zinc centers here, zinc ions here, that's the core of the active site. And what we saw is there's this blue surface here, this kind of groove, and we thought, you know, maybe if you stuck some piece of protein like a helix in there, that might block up the active site and prevent it from being active. But there was immediately a problem with this, and it was that the termini of CpG2 were not even close to the active site. So as a first step, we needed to move the termini of CpG2 to a location closer to the active site by making a circular permutation of the enzyme. And I'm not going to dwell on this too much, except to say that we did it and we were able to get enzyme circular permutants that are essentially as active as the wild type enzyme. So that being done, we took a look at our active site and said, okay, let's try, you know, kind of the most straightforward approach that we could, which is just to dock in a simple helix, build an interface here between a glutamate residue and the zinc to kind of fill in the zinc complement coordination sphere, build an interface using design, and attach the whole thing to the protein using a linker, which has that protease sequence coded, the cleavage sequence of a protease coded into it. And so we tested that out in the lab. We tested a few of these, and we were actually able to get an enzyme. So this is the wild type enzyme here. We inhibit it with the prodomain. We see the drop in activity. And then when we incubate that with the protease, here the matrix metalloproteases, which were coded into the linker sequence, we see the activity comes back. So that was pretty exciting. We actually were able to reversibly inhibit the enzyme. But the amount of inhibition that we were getting was less than what we were hoping for. We wanted a bit more of a dramatic decrease. And so we moved on to this massively parallel approach. And so we basically looked at the characteristics of the helical prodomain, which we thought were hopeful. Namely, we tried attachment to both the C and N terminus, the new C and N terminus, and the C terminal ones worked and the N terminal ones seemed not to. So we decided to stick with that C terminal attachment. And we also kept that glutamate that I showed you coordinating the zinc as a central part of our design strategy. And so we designed a library with about 15,000 pro-enzymes in it. So those were, after design, we had 15,000 of them. The original source of these small prodomains that we were using were, first of all, small domains that we grabbed from the PDB, and we complemented that with the de novo designed mini proteins that Gabe Rockland designed in the Baker lab, and those had been confirmed to fold using a proteolysis assay. So we took advantage of that to use those well-folded ones from his study in our study, and that gave us several thousand more mini proteins to work with. So the way our design strategy works was basically we built an inverse rotamer library of glutamates, as you can see here, coordinating this zinc. So this basically enumerates all the backbone positions that you can use to get coordination between a glutamate residue and zinc. So if you put a backbone residue overlapping with one of the sets of backbone atoms from this library, then that should allow you to form that interaction. And along with this, we superimpose, you just keep throwing these mini proteins at this inverse rotamer library until you find one that matches and doesn't clash with the rest of the protein. And so once you have that, we hook up the end of the terminus of the enzyme to the terminus of the prodomain. And we did some sequence design in order to build a nice interface between the scaffold and the prodomain. And I'll note that this was one-sided design. So we didn't change the sequence at this stage of the enzyme because we didn't want to make a whole lot of mutations that were going to reduce the activity of the enzyme in the active site vicinity. And then, and so then out of that, we ended up with about 15,000 prodomains, and I'm showing 12 of them here, so you can see that they are fairly varied in terms of their structure and the orientation in which they bind. And so then we needed some way of taking that 15,000 and screening them. And so I started off by just using a computational metrics to bring that down to about 7,500. But obviously I can't make 7,500 designs in the lab all by myself. So we needed some way to get down to a more manageable number, like say 12. And so in order to do that, I designed an assay that allowed us to screen this entire library at once. So this took advantage of the ability of the enzyme CpG2 to cleave and detoxify the drug methotrexate. So if you take E. coli and you subculture it into media containing increasing amounts of methotrexate, you will see that the E. coli won't survive as the amount of drug increases. So the more drug it is, the less likely you are to get that culture to grow. If, however, that enzyme is now expressing CpG2, or that E. coli is expressing CpG2, the E. coli will now grow robustly regardless of methotrexate. So it's able to detoxify the methotrexate and survive. Now if we take that CpG2 and we add a prodomain to it, as you can see here, and let's say this is a poorly inhibiting prodomain, a weak prodomain, which I'm denoting in green, and we're going to say that a green prodomain is weakly inhibiting and a red prodomain is strongly inhibiting. If we do that we would expect to grow just like regular CpG2 because the activity is not impeded, so we're still going to get the detoxification of that drug. Now let's imagine we have that first generation design, that helical design, which has a moderate decrease in affinity as compared to the wild type. In that case, we would expect lower concentrations of CpG2, it would still be able to, or of methotrexate, it would still be able to survive. But as we get to higher concentrations, the survival starts going down. So we're now selecting for against those a little bit. And if we have a strongly inhibiting prodomain, in this case, we would expect that the enzyme, the E. coli doesn't survive well, similarly to the E. coli without any CpG2 at all. So you end up with nothing really growing except maybe at very low methotrexate concentrations. Now let's think about it if this E. coli is expressing our library. So you've got lots of E. coli, each expressing one gene, and this library spans the entire spectrum from green to red. So we've got everything there. If we take that and put it in the culture without methotrexate, we would expect to recapitulate the entire library. And as we increase the amount of methotrexate in our culture, we would expect to push it more and more towards the green designs, the ones that are still highly active and are able to detoxify the methotrexate. Now, you may have realized that I just told you that I designed an assay that selects for weakly inhibiting prodomains, and we want, of course, strongly inhibiting prodomains. So that was a bit of an issue. And so in order to resolve that, we use next generation sequencing to profile all of these cultures in order to see which prodomains were disappearing and by extension, which ones were the ones that were most strongly inhibited. So starting from our 7,500, we identify about 1,400 of them that had high quality sequencing data. And this was really important because of this negative selection scheme. It was important that we only use the ones that had high quality sequence data across all cultures and in order to make sure that we weren't just picking up noise. From that 1,400, 108 of them, their behavior in NGS was consistent with what we expected for a pro-CpG2, and that does better than the helical domain, the helical prodomain that we had identified at the beginning. That was kind of our benchmark, our control. And of that 108, 27 of them we identified as being like high probability pro-CpG2. So those were the ones that seemed to have the best NGS profiles according to what we were seeing. Of that 27, I looked at all of them, gave them the eyeball test, and picked 12 that were significantly different from each other and looked good to me. And so these are that 12 I showed you before happened to be that 12. So you can see, once again, they're quite different from each other. So we're picking up different kinds of structures here. And yeah, so we went ahead and tested those in the lab. And what we saw was, here's regular CpG2, here's our benchmark, the helical prodomain. And if you take a look, all 12 had lower activity than the helical one, some of them by a fairly significant margin. So we were pretty excited by that. We wanted to make sure that this wasn't just that, most of these prodomains were inhibiting, and that it was just, we just picked 12 and it was kind of, if we had picked any 12, we probably would have seen the same thing. So we checked 4 that were predicted to be poorly inhibiting, and these have activity that is at or near wild type levels. So we are able to distinguish between the good and poor prodomains. And when we take this and stick it in with a protease, we see that, similar to the helical case, we see the activity comes back. So we are able to recover that activity by incubating with a protease. So these are true pro-enzymes, they're not just broken enzymes. So, and then going back to our original scheme, this means that if you were to give a patient this enzyme, it would travel through the bloodstream with lower activity, arrive at the tumor site, get cleaved by a protease, turning on the activity and allowing it to trigger that toxicity, more specifically at the tumor site than in the rest of the body. So it was nice to show this in E. coli and whatever, but we wanted to see if there was any evidence that this has any kind of, if this difference in activity is enough to actually see anything. And so we weren't able to do this in animal models or anything like that, but we were able to do it in a PC3 cancer cell line. So PC3 cells are an aggressive prostate cancer cell line. And so we basically tried to see if there was a difference in the activity that we could detect by the cell survival in these PC3 cells. And we can see that over a certain concentration range, we do get a difference in activity between the circular permutation and the pro-enzyme. Okay, so having done this, I'm, by training a structural biologist and enzymologist, so I'm really interested in how are these things working from a structural and a enzymology kind of perspective. And so first thing we wanted to do, we weren't able to get crystal structures of these. And so we wanted to see if we had any evidence that our model was actually correct. And so to do that, we added a disulfide bridge between the prodomain and the scaffold. And that disulfide bridge allows us to, if our model is correct, that disulfide bridge should covalently hold that prodomain in place, and we would expect that that would help push if there's some equilibrium of that prodomain coming on and off of its correctly bound position, we would expect that that would really restrict it back to the bound form. And so we then did Michaelis-Menten kinetics to look at what the source of this inhibition was for both the base designs and the inhibited designs. And so this here, so if we look here at the K-cat values, we can see this is the circular permutation. And you can see that all three of our top designs in the red bars had diminished K-cats, indicating that the catalytic turnover rate was slowed. So the actual machinery of the enzyme was being slowed down. And the disulfide variants do have a further decrease in activity from there. And then we looked at the Km, and here you see in 2 of the 3 cases, we see an increase in Km in the base designs. The third one actually gets a little bit lower, so we're seeing, and this is measuring basically the affinity of the substrate to the enzyme. So in these cases, we're seeing a slight reduction in affinity, and here we're actually seeing that the affinity increase a little bit. The disulfide variants in 2 of the 3 cases were moderately higher, and in 1 case it got a lot higher, so that's what we saw there. And so then when we look at the overall catalytic efficiency, we see that we get a pretty significant drop in the activity as based on the K-cat over Km. So then we wanted to know, so the Km is kind of perhaps self-obvious. If you're sticking this protein in there, you might say, well, okay, so that's going to block the active site just sterically, and that's why we see a decrease in Km, or an increase in Km, a decrease in affinity. But why do we see that decrease in K-cat? What's the mechanism behind that? And so this we turn to small angle X-ray scattering, which gives us a low resolution view of what our enzyme is doing. So I haven't shown you this before, but CpG2 is a dimer. So this is the one protomer, the other protomer. And it is believed that CpG2, as part of its catalytic cycle, undergoes this conformational change from the open conformation, which is thought to be responsible for substrate binding, and the closed conformation, which is kind of when the conformation when activity actually happens. When we looked at the SACS data and modeled it as a 2-state equilibrium between these 2 states, we see that in the absence of substrate, we see roughly a 50-50 mixture of the 2 components. But when we add substrate in, that pushes the equilibrium strongly towards the closed conformation, 75 versus 25. And so that kind of makes sense. You put the substrate in, it binds, and then the enzyme closes up on that substrate in order to catalyze the activity. Now let's think about what happens if you put that prodomain in there. With the prodomains there, the enzyme is not able to adopt the closed conformation. So what would need to happen is that the prodomains would have to come off of their standard, their position, and allow the enzyme to close up, as you can see here. And so we might expect that if those prodomains are bound, it would diminish the ability to adopt that closed conformation. So we did again that 2-state modeling, and what we see here is that although with the substrate present, we still see that 50-50 mix. You might see a slight preference for it in design 1 for the open conformation, but not a whole lot. In the presence of substrate, that equilibrium did not move toward that closed conformation. So it remained in that sort of 50-50 regime. It didn't move towards that 75-25. So we think this is evidence for the prodomains basically impeding the enzyme from getting to that closed conformation, and that would at least partially explain why the K-cat slows down when you add these prodomains in. So to summarize that story, and I'll maybe pause for questions after this before I move on to the next, how much time do we have? Oh, we've got lots of time. So to summarize, I've developed a computational method to build an auto-inhibited form of CpG2. I used a high throughput screening approach to select for the most strongly inhibiting variants. and showed that those variants can have their activity restored by incubating with the appropriate protease. The difference in activity is sufficient to show differential activity in cancer cell lines, as I showed you with the PC3 experiment. And the kinetic and SAX data highlight some of the possible mechanisms of auto-inhibition that may be at play with this system. And so that was all I wanted to say about that story. So before I move on to the other part, if anyone has any questions about that first part, I can take them now, or I can take them at the end. I don't know.
Yeah, that's an awesome story, Brahm. I'd be happy to ask questions now if other people are interested, and then we can have dessert afterwards with the.
Yep.
Nice. So maybe I can just jump out and lead the questions here. So it's an awesome story. So many cool technologies that you had to develop along the way. And the first one that was really striking for me is your platform for screening such a large number of designs seems really powerful. So just wondering if you had thought about taking a next step, maybe take your lead design and then build a mutational library in the surface or just in the pro-domain, and then you might be able to select for something that's even tighter out of that. Yeah, so I think I think that's definitely makes a lot of sense as a next step. So one of the challenges of this particular screening system is that you can't really, so what would be really ideal is if we could select on both, if we could do two-sided design basically. So if we could start really adding a lot of mutations in the scaffold portion, so that would be nice. One problem with that is that it would be very hard to distinguish between mutations that just on the scaffold side abolished activity and just so not related to the prodomain, and especially because this is binding at the active site, so we expect a lot of these residues to be important, and actual residues that are reducing the activity because of that interface. But I think certainly there's a lot of potential to do more design on that prodomain using this kind of approach, and I think that's in the works. So yeah. I think Per has a question too. Per.
Yeah, thank you, Brahm, for a very nice talk. I have three small questions. So one is regarding the enzyme. I'm not quite understanding why do you actually need the enzyme and not have a substrate attached to an antibody, which you would normally do currently. So the upside of that.
Yeah. So one of the big advantages of using an enzyme rather than so that's kind of antibody drug conjugates is that enzymes are you are able to get catalytic amplification using an enzyme. So if you're thinking about a antibody drug conjugate, you're going to get, if you have one molecule of drug, you're going to have, it's 1-to-1. Whereas with an antibody, if you've got or an enzyme, I should say, if you have an enzyme sitting on the tumor surface, you can have one enzyme that can turn over hundreds or thousands or, on and on drug molecules. So you can get a very, very strong response from a very small amount of
But didn't you bring the substrate along with the enzyme?
No, so the enzyme is administered separately, and it just goes through the system. So it's free to do what it wants.
Gotcha.
Yeah.
Then I have two small questions regarding, so one is the basal level in your protease assay. So don't you see any degradation of the enzyme itself there?
No, so the proteases are fairly specific. That it's not, you were not talking about like the MMPs, so they're they're not quite as stringent as something like a TEV protease or something like that that we use commonly in the lab, but they're pretty specific, so they're not going to cleave well-folded proteins. So there might be some degradated, but it's going to be very, very small, and mass spec kind of shows that we don't really see any significant fragments after incubation with the protease.
Okay, and then last but very short, so when you did the disulfides, if you did the enzymatic assay in a reduced in a reduced environment, did you see the activity actually go back then?
Yeah, we were really hoping we could do that experiment. The challenge with that is that I think as a consequence of it being a zinc enzyme, if you put it in a reducing environment, it just basically, the whole thing goes, like the activity goes way down and we can't really see anything clearly. So unfortunately that experiment wasn't possible.
Okay, thanks.
Yep.
Awesome. Other questions for Brahm at this stage here?
I can stop at the end also, and if we can come back to the first part if people have other questions that they think of in the meantime. But yeah, I can move on. So this next is a bit more of you can think of this as a bit more of a sales pitch. If anyone here actually is working on protein therapeutics, it's less applicable to antibody therapeutics, which ironically is what I'm working on now, but it certainly is applicable to things like enzymes or other regular proteins, non-antibody biologics. And so this is a feature we added to Rosetta that allows you to easily de-immunize protein therapeutics as you design them. So as many of you I'm sure are aware, foreign proteins that you might deliver as a drug are immunogenic. So that means that, so this over here on the right is the MHC2 molecule, and it has this peptide that it grabbed from some protein that it decided was immunogenic, and it displays this protein and alerts the immune system that this is foreign and that we should destroy it. And so if you have this situation arise with your biologic, the kind of best case scenario is that the immune system just identifies it, clears it out of circulation, and neutralizes it. So that's kind of like the good story. The worst case scenario is that this actually triggers a systemic immune response in the patient, and that can be actually quite dangerous. So this is a serious problem anytime anyone is dealing with a biologic drug. And just to give you an idea of the scale of this problem, here are a few, numbers are a few years old, but a few biologics that are either in the pipeline or on the market. And if we take a look at the percentage of patients that have anti-drug antibodies, so this means that they are having some kind of immune response to these, you can see that these numbers, depending on the study and the particular enzyme, these numbers are quite high. So that means that this gives you some insight into how serious a problem this can be. So it should come as no surprise that people have tried very hard to come up with ways to identify which specific portion of your protein is immunogenic, which one is going to be displayed, like I showed you on that first slide, on the MHC2 receptor. And I'm going to focus today, well, I'm primarily going to focus on a predictor called ProPred. And while this is a fairly old predictor, it was published in 2001. And it's very straightforward in how it works. It uses a scoring matrix. So if the core peptide that's displayed on the MHC2 molecule is a 9-mer. And so that core peptide gets displayed. So that's the core that we're looking at, that 9-mer. And then you basically see what residue at each position, 1 to 9, and you get a number associated with each thing at each position. and you add up those scores, and that whatever score you get there determines whether or not your peptide is a binder or not. And so this is very, very fast, but not the most sophisticated thing in the world, and we know that our immune system is far more complex than that would indicate. The other one we looked at is the net MHC2 predictor, and this is kind of your state-of-the-art epitope predictor. It's here you can see published in 2018. This is using machine learning methods in order to predict whether or not something is an epitope or not. And so this is a much more sophisticated predictor, but it's much slower. And you'll see why that is important in a second. So how would you normally go if you have, let's say you've got your molecule that you think has potential of being a biologic? Normally, what kind of approach would you take if you want to de-immunize it? So first thing you might do is take that sequence and predict where your epitopes are based on a predictor like those two that I just mentioned. And so you figure out where your predictor, where your epitopes, your key epitopes are, and then you maybe use evolutionary-based PSSMs or some other kind of way of coming up with a list of mutations you want to try that you're hoping will knock out that epitope without altering the activity of your enzyme. And that's really important in here is that we don't want to, unlike most design problems, we don't actually want to change the activity in this case. We just want to tweak the sequence a bit and have like kind of conservative, evolutionarily conserved or otherwise conserved mutations. And then you might take your list of mutations and try some structure-based validation using a tool like Rosetta or something like that to see which ones you think will be well tolerated from a structural point of view, and then go back and do more epitope predictions on the ones that the combination of mutations that rank well, and see if you knock down your immune score, and if you need to do more de-immunization, in which case the process starts over, and you keep doing this over and over again. And so while that's fine, it's kind of not the most straightforward approach, and certainly it doesn't really lend itself well to a case where you're actually designing your protein from scratch rather than just using an existing protein to do this, to act as your therapeutic. So we developed this score term in Rosetta called MHC epitope energy. And what this is supposed to be is an integrated computational de-immunization approach. So the MHC epitope energy is going to take care of determining what peptides in your protein are immunogenic. You are able to use this with the Rosetta design. So anytime you're using Rosetta, MHC epitope can be used. So it will work with your design protocols. And if you want to use PSSM-based filtering, similar to keep your design space in kind of a conservative space, you can still use that because that's possible to do within Rosetta using res files and things like that. And we actually added some tools to help make those res files if you want to use them. So the way this works is MHC epitope energy basically slides, it has a window in it, slides through sequence space and scores your peptide using a predictor. So you go through and you look at each peptide and you ask how many of your MHC2 alleles bind to this peptide. Once you get to the end of your sequence, you add those up, and that becomes your MHC epitope score. And these scores are all cached. So when we run the packer during design, rather than recomputing the entire sequence each time, we only recompute the peptides that have changed as a result of the substitution you're considering. And so that speeds up packing quite a bit. And so you don't see a big slowdown as compared to just running the packer without this score term turned on. So let's say you think this is useful for your work. How do you use it? The basic interface is actually really easy to use. So let's say you have a Rosetta script. You define, this is a very simple Rosetta script. You've got a score function, you've got a mover, and you run that mover. So this is your standard protocol. Let's say you wanted to turn it into a de-immunization protocol. All you have to do is turn on the MHC epitope weight by setting it to something other than 0, and then point it to a configuration file. And we've included this ProPred 8.5 MHC configuration file in the Rosetta database, which means that it will just start using ProPred and try and minimize that ProPred score without any other modifications required on your part. And this should in principle work with any design protocol that you are that you want to run. Having said that, we do have a few recommendations. We recommend that you use a weight of 1. That seems to be a reasonable balance between getting reasonable Ref 2015 scores and pretty reduced ProPred scores. We also suggest that you restrain the number of positively and negatively charged residues, and that you do that separately, that you count each one separately rather than looking at the net charge. And this is because ProPred and epitope predictors in general like to put aspartates in because they tend to not be very immunogenic. So if you don't want your protein to turn into polyaspartate, it's a good idea to limit the number of negatively charged residues that you're adding and positively charged residues as a consequence. And we also recommend that you use those evolutionary constraints or some other way of limiting your design space to more reasonable mutations. And not only does this mean that you're going to more likely have mutations that lead to an active protein, this can greatly reduce the runtime because you have a smaller design space to sample. And so this can take something that would normally take like an hour to run down to like 10 minutes or something like that. So that can be very helpful in that respect as well. Now, I've been talking about ProPred a lot, and you may be wondering, well, you said this is a 20-year-old method that you know is not the gold standard anymore as far as this stuff goes. So does this actually do the job? And so we took our designs and de-immunized the ProPred de-immunized designs, and we scored them using net MHC2. And what you can see here is that there's a reasonable correlation between ProPred and net MHC2, especially actually if you use a stronger threshold, a more stringent threshold from your evolutionary PSSM. But in general, the ones that have low ProPred scores, you will see some that have a low net MHC2 score. And while you're saying, well, how do I know that the one I'm looking at is not one of these? And the good news is that you don't actually have to count on this. You can take all of your designs and score them after the fact using net MHC2. The point here is more that it is reasonable. If you de-immunize with ProPred, you are going to get designs that look good according to net MHC2. So this is, even though it's not the best predictor out there as far as sophistication, you may be able to get most of the way there just using ProPred. And it's a good way to go, especially if you're just trying to keep, if you're designing a new protein and you're trying to keep your immunogenicity low. Yeah. So we also have added some more customization in order to deal with problems that may be a little bit more challenging and you need a higher level of sophistication. One of them is to allow your de-immunization to target specific hotspots. So this prevents you from adding mutations all throughout the protein and just focuses on, so if this happens to be CpG2 here, and you could say there are certain hotspots that are really the ones that maybe you want to focus on. And so you can define constraints that say, I really only want to look at de-immunizing these particular regions rather than looking at the whole protein, and that helps limit the mutational load and have designs that have fewer mutations, more likely to be active, I guess. Another way of doing this is to threshold the score. So you can say, rather than trying to bring everything down to 0, let's just bring it down to this limit that you can set and say, okay, I'm only going to, if it's above this line, we'll try and reduce it. If it's below the line, then don't worry about it. We'll just leave it as is. And we've also added an ability to use net MHC2 in a limited way by pre-computing. For this to work, you need to limit your design space using a PSSM and probably also using constraints. But you can basically, if you have a small enough design space, you can enumerate the peptides that you're likely to encounter in your protein and pre-compute all those scores and put them in a database of NetMHC2 scores and just query that database rather than running NetMHC2 itself. So that's a way of de-immunizing using NetMHC2 if you want to take advantage of that in that more powerful predictor. We've also integrated this with the NMER-SVM approach. So the existing way of de-immunizing proteins in Rosetta was using this NMER score term. The main difference between what we're doing and that score term is that that score term was not Packer compatible. And by plugging it into our interface, we are now able to use that score term with the Packer. And so you have kind of like a one-stop shop to whatever predictor you want to use, whether it's NMER or one of the ones we've implemented. That's all available through the NetMHC2 interface. And Indigo King, who is the one who introduced this score term originally when in the Baker Lab, I believe is using our interface at Cyrus to run that the SVM-based approach. So I think that speaks to that this is hopefully a nice way for people to do this kind of work. It's well documented, so if you have questions about how it works and what to do, you can see that this is available on the Rosetta Commons wiki, and it is also part of the scientific test suite, so we're testing regularly to make sure that we're getting reasonable designs out of our as Rosetta changes. So to summarize, I've developed a score term in Rosetta that allows you to de-immunize proteins while doing design. And you have a very simple interface if you want just a straightforward de-immunization problem, as well as a lot of customizations if you have a more challenging design problem. And it's part of the Rosetta Scientific Suite, so yeah, so that's being tested in that way as well. Last but not least, the acknowledgements. I'd like to first of all thank Sagar and all of the past and present members of the Beccari Lab for their help and support on these projects. I'd like to mention in particular Kristen Blacklock, who worked on another aspect of the DEPT story that I didn't mention, which is to make a light activatable form of a DEPT enzyme. As well as Laura Azuz, who was a Rosetta intern who worked with me in 2016, and she actually did the original computational design of that helical design. So thank you to her for doing that. I'd like to thank Justin Drake's group at University of Minnesota, and specifically Ralph and Victor. They're the ones who did the PC3 experiments in cancer cell lines, so big thank you to them. Moving over to the de-immunization project, I'd like to thank Chris Bailey Kellogg, who is a prof at Dartmouth, and we work very closely together for implementing that score term in Rosetta, and he's done a lot of work on de-immunization in the past, so it was very nice to rely on his expertise. And Vikram Mulligan, who now at the Flatiron Institute, who a lot of the infrastructure that we used in that score term was originally developed by him. So without his work, there's no way we'd have been able to do what we did in the way we did it. So big thank you to him as well. And he also helped us out making sure our code was up to standards. So thanks to him. And funding agencies for funding, and all of you guys for your attention in virtual land. And if there's any other questions about any of this, I can take them now. And yeah.
Awesome. Really exciting stuff, Brahm. Yeah, let's throw it open for questions.
I had a quick question.
Sure.
Thanks. Maybe just a novice question, but and I'm not really familiar with ProPred. How do you approach the MHC allele question? I think you mentioned some alleles. How do you approach the vast numbers of HLA alleles, or maybe that's a ProPred question?
Yeah, so that is something that is dealt with. So ProPred basically has a different, so I mentioned that it's a scoring matrix, so it's a 9x20 scoring matrix, which you use to rank position by position what score is what, and then you basically add that up to figure out whether or not you have a binder. Each allele has its own matrix, and so what we are getting at the end is the sum of the, so if a particular peptide binds 2 out of the alleles that we tested, that peptide will get a score of 2. And then you sum up all the peptides in your protein to get the total score. And so that's basically how ProPred does it, and so we basically just use that. And NetMHC2 is going to have a similar kind of philosophy in that it's going to rank each allele. Each allele is going to be evaluated one by one and then added together.
So it's looking at a kind of a cross-section of 20 major HLA alleles. Did I get that number right?
So the ProPred, our implementation of ProPred uses, I think it's 8, which is selected. It's based on, there's some papers that look at which combinations of alleles give you a good cross-section of it's kind of which alleles are important and cover, kind of the world population reasonably well. Then MHC2, there's several different options. I think there's one option that's 7 alleles, one that's 27, and you can look at the entire set that they give you as well. So there's different ways of doing that. And those are things that you could customize as well, depending on what you're particularly interested.
Nice. Thank you.
Yep.
Maybe I can ask the obligatory antibody question. So have you tried using this on immunogenicity predictor on CDR loops?
So no, we haven't, and Chris would probably be the better one to ask this kind of question to. But I think and so I can't give you a really great answer to it. But I think the bottom line is that these predictors work. They've kind of been trained on a general case, and antibodies are kind of special, and there's a better, they're not necessarily, the results you would get would not be as meaningful. And all of this stuff, like computational prediction of epitopes is not it's not a straightforward thing to do to begin with. And so I think when you put it in an antibody context, I think these sort of conventional humanization approaches, which are just like kind of mutate things to be back towards the human germlines, are going to give you a better bang for your buck than trying to get these predictors to produce a good way of de-immunizing your antibodies. So it really is more valuable for everything except antibodies, which is of course the biggest one in the field, but maybe that will be something that's going to be changing in the as we go forward, especially as protein design tools become more sophisticated.
But just to actually expand a little bit on Chris's question here, one of the issues with antibodies is that it's self, meaning that it's recognized by the immune system as not being foreign. So it would actually probably be quite easy to have a subtraction term where you just subtracted anything that is actually considered human. Is that something that you could actually do easily with the setup you have?
I think, well, I mean, I guess, so I mean, it would not be that hard to build in something that said, so if you want to add a new predictor, which is essentially what we would be talking about here, you basically just need something that takes a peptide, does some calculation on it, and returns a score. So if you had something to say, here are the kind of human sequences, and we're just going to compare your sequence to those human sequences, that might be doable. I think the typical antibody workflow, as I understand it, is you typically just take your antibody and you align it to human germline sequences and then start making some reversions. And that's probably a smaller problem that can be dealt with reasonably efficiently, you know, in a kind of direct way rather than...
No, I completely agree with that assessment, and that's also correct. But I'm thinking about CDR3, right? CDR3 will always be foreign. Mm-hmm. That's the one you actually want to use this method on, in my opinion.
Yeah, yeah, so I mean, incidentally, for something I'm doing at work now, I actually did end up just to restrict the number of sequences I was looking at. I did actually use this as a filter to throw out sequences that show binding in this way. I couldn't tell you if that's effective or not because I haven't gone ahead and tested those yet. But it is certainly an interesting idea, and it's certainly doable with things as they are now. We have tools that let you evaluate these kind of outside of Rosetta as part of the Rosetta tools package, and you can also, if you're doing design in HCDR3, you can certainly turn on that score term and it will work. I can't tell you if this is the best approach for doing that, but certainly it's an option and would be very interesting to see if that does help.
Yeah, it'd be interesting. Very nice. Thanks.
Any other questions for Brahm tonight? All right. Well, Brahm, thanks again.
Thank you.
It was two awesome stories. Great to see everybody.
Thank you.